Hi everyone, this is Dr. Minoy Cristobal from the Dr. Stein Monogastric Nutrition Laboratory. Today I will be discussing one of our experiments titled Feeding intact protein from soybean meal instead of synthetic amino acids does not affect growth performance, carcass composition, or energy deposition by growing pigs. This research was presented at the ASAS Midwest meeting in March of 2024. The industrialization of some key amino acids led to the development of the amino acid industry. For instance, the first amino acid that was industrially produced was methionine in the 1950s, followed by the commercial production of lysine in the 60s. A few decades later, the industrial production of threonine and tryptophan started, and from around 2015, all indispensable amino acids are available in a synthetic feed-grade form. Diets for growing pigs based on soybean meal and cereal grains typically meet amino acid requirements and maximize growth performance and protein synthesis. However, the growing demand for more precise nutrition has led to increase the use of synthetic amino acids in pig diets. Over the past few decades, the use of synthetic amino acids has increased while the inclusion of soybean meal has declined. With less soybean meal inclusion in diets, crude protein is also reduced. The effects of reducing crude protein in diets for pigs has been addressed by several studies. In general, the studies reported a lower nitrogen excretion, no negative effect on growth performance, increased net energy of diet, reduced incidence of post-weaning diarrhea, and lower feed costs. However, some studies also reported a negative effect on nitrogen retention, more fat and less muscle deposition, and changes in intestinal morphology of pigs. In this study, we focused on the effects on growth performance, net energy of diet, nutrient deposition, and carcass composition. Regardless of whether amino acids are supplied by soybean meal or synthetic sources, it is generally assumed that protein and energy deposition occur at similar rates as long as amino acid requirements are met. However, results of recent research indicate that daily nitrogen retention might decrease when pigs are fed diets with less soybean meal and more synthetic amino acids compared with diets containing more soybean meal. In a previous video, we discussed the effect of reducing dietary protein in corn-soybean meal diets supplemented with synthetic amino acids on nitrogen balance, and we observed a linear reduction in daily nitrogen absorbed from 30 to 20% and a linear reduction in daily nitrogen retained from 23 to 17%. This may indicate either an amino acid deficiency in diets with synthetic amino acids or that there are other factors in soybean meal beyond amino acids that are beneficial for pigs. Therefore, the objective of this experiment was to test the hypothesis that reducing crude protein by reducing the inclusion of soybean meal while increasing inclusion of corn and synthetic amino acids will not increase diet net energy and will not affect growth performance, carcass composition, or protein and energy deposition by growing pigs. Moving on to materials and methods, we formulated 4 diets. Diet 1 contained 60% corn and 35% soybean meal, some soybean oil, and no synthetic amino acids. And this diet had 20% crude protein. Diet 2 was similar except that we included 3 synthetic amino acids, and with that, soybean meal inclusion was reduced as much as possible, but to still meet the requirements of digestible indispensable amino acids for growing pigs. This diet had a crude protein of 16%. For the next diet, we followed the same logic, but with the inclusion of 4 synthetic amino acids, and crude protein of diet was reduced to 15%. The last diet included 5 synthetic amino acids with a crude protein of 13%. In the last 3 diets, the synthetic amino acids were added in the order of most limiting amino acids for growing pigs, and as we reduced soybean meal inclusion, we increased corn inclusion. A total of 176 growing pigs of around 32 kilos were used in the experiment. Among these, 16 pigs were randomly selected at the start of the experiment and designated as the initial slaughter group to establish a baseline to determine nutrient deposition. The remaining 160 pigs were allotted to the 4 diets using a randomized complete block design. Thus, there were 4 pigs per pen, 2 gills and 2 barrows, and 10 replicate panes per diet. The data was analyzed using SAS, and the fixed effect was the diet and the random effect was the replicate. Contrast coefficients were calculated to determine linear and quadratic effects of dietary protein. Diets were provided to pigs on an ad libitum basis for 28 days. At the conclusion of the experiment, one gilt or barrow was randomly selected from each pen for a total of 20 gilts and 20 barrows. 2 blood samples were then collected from the jugular vein of each selected pig. After that, the selected pigs were transported to the Meat Science Laboratory at the University of Illinois and slaughtered after an overnight fast. Samples of organs, including intestines, were collected and carcass were processed. Moving on to the results are presented by different colors from left to right, from the 20% crude protein diet to the 13% crude protein diet. Final body weight of pigs was not affected by dietary treatment, and average daily gain was not affected by reducing soybean meal and increasing synthetic amino acid in diets. Likewise, average daily feed intake by pigs were not affected by reducing soybean meal and increasing synthetic amino acid in diets, which resulted in no effect on gain to feed ratio. Hot carcass weight and dressing percentage were also not affected by dietary treatments. The lack of differences in growth performance is likely a result of the fact that reduced protein diets supplemented with synthetic amino acids can provide enough nutrients to meet minimum requirements by pigs to maximize growth. It is therefore likely that the amino acid profile in the diets fed in the present experiment was closer to the requirements of the pigs than some previous experiments where reduced growth performance was observed in pigs fed low-protein diets. The current data are in agreement with data demonstrating that it is possible to feed reduced protein diets without negatively impacting growth performance or carcass characteristics. To measure retention of nutrients, we use the initial slaughter group and we determine the nutrient composition in the body of those pigs to establish a baseline, and we consider the nutrients retained, the nutrients that were deposited on top of that baseline. This is an example for retention of crude protein, but let's take a better look at these values. We observed that retained protein and retained lipids in grams per day were not affected by dietary treatment. Likewise, retained energy in megacalories per day was not affected by dietary treatment. Then we calculated the retained lipid to retained protein ratio for each diet, and we observed that the values were close to 1, indicating that pigs deposited similar amounts of lipids and proteins. And there were no effects of dietary treatment on the retained lipid to retained protein ratio. The lack of differences in retained protein and lipid among pigs fed experimental diets contrast with results indicating a reduced protein retention and increased lipid retention in pigs fed low protein diets compared with pigs fed diets with greater protein concentration. Results from the present experiment were also different from data indicating that energy retention is increased in pigs fed low protein diets. We also observed that energy efficiency tended to quadratically decrease as dietary protein was reduced. In this graph, we compare the calculated net energy using NRC values in dark blue with the determined net energy in diets from the current experiment. We observe that the calculated net energy seems to go up, whereas the determined net energy seems to go down. It was expected that net energy would increase as dietary crude protein decreased because the net energy in soybean meal is believed to be less than in corn. Therefore, reducing soybean meal and increasing corn concentration should have increased net energy. Furthermore, if reducing soybean meal increases diet net energy, then the gain to feed ratio should have improved as well. However, the observation that no increase of net energy in diets or improved gain to feed ratio was observed as crude protein was reduced in diets indicate that net energy in soybean meal is close to or greater than the net energy in corn. In fact, we observe a tendency for a reduction in the determined net energy as soybean meal in diets was reduced, indicating that the net energy in soybean meal might be greater than in corn, which has also been reported by other experiments in which net energy in soybean meal was estimated from growth performance of pigs. In other experiments in which net energy in soybean meal was determined using indirect calorimetry, the net energy in soybean meal was close to the net energy in corn. These observations implied that current book values might underestimate the net energy in soybean meal. There is therefore a need for generating new prediction equations for net energy in diets for pigs, mainly due to changes in animal genetics over time and changes in soybean meal composition and quality. Based on these results, we conclude that the hypothesis that reducing diet protein concentration by reducing dietary soybean meal and increasing corn and synthetic amino acids would not increase diet net energy was confirmed, as results demonstrated that there was a tendency for a reduction in net energy as crude protein was reduced. Likewise, the hypothesis that reducing diet crude protein would not impact growth performance or carcass composition of pigs was confirmed. Overall, these results indicate no advantage of reducing diet soybean meal concentrations, but the observation that the net energy in soybean meal is likely greater than current book values deserves further investigation. With that, I would like to acknowledge the United Soybean Board for its support in this work. And also thank you to all Dr. Stein Monogastric Nutrition Laboratory members for their help. And thank you for your attention. If you would like to learn more about our research, please visit our website.